Hi everyone, thanks for joining us today. My name is Sarah Schuett and I'm a software sales specialist with Beckman Coulter. And today I'm going to be talking to you about the Cytobank platform and the machine learning algorithms that are found within it. So this is the first session of our bootcamp that we're running throughout the month of April. Thank you so much for attending. If you have any questions today during the presentation, please feel free to use the question and answers box. And we will do a live Q&A at the end where we'll address all of those questions. All right, so to get started, what is the Cytobank platform? So the Cytobank platform is a cloud-based platform that we're going to use to analyze our data. What that means is after we acquire our FCS files, we can send them to the cloud and we can use the cloud to process all of the data analysis we're trying to do. And that's really important when we talk about these machine learning algorithms because they're going to be rather resource intensive. Also, because we have everything fully integrated in the platform, we're not going to have to worry about installing plugins, and we're not going to have to worry about knowing R to access these algorithms. Everything's going to be fully integrated within the Cytobank platform. And then since it is a cloud, we also have analysis and data management and data archiving capabilities within the cloud. So once we have everything uploaded into the cloud, we can go in there and we can manage how our data is displayed. We can organize all of our different experiments, and we have the ability to archive all of that data to keep track of our data over time. And the great part about being a web-based software, especially right now in the current situation we're in, where we're all having to adjust to a new normal of working from home or working very briefly in the lab, is that everything's going to be centrally located on the cloud. So all of the data that's been uploaded will be available on the cloud, and all of the analysis that has been completed thus far will be uploaded onto the cloud. What that means is we can access that anytime, anywhere from any web-enabled device. So if I wanted to get on my iPhone, I could go ahead and grab my Cytobank experiment on my iPhone. I can access it from my iPad, from my laptop, from my PC, and I'm going to be able to store, share, and do analysis on that data anytime, anywhere. And because we're in the cloud, what it's going to do is it's going to allow us to do different analysis runs at the same time. So when we talk about these algorithms, they're going to be computationally heavy. And so what we can do is we can run up simultaneous runs and have those runs executed at the same time, so we can save ourselves time on the back end. We're also going to be able to do project collaboration as well. So once it's within the cloud, we can share that experiment with collaborators, maybe at our institution, maybe outside of our institution and around the globe. And it's really going to allow us to focus on collaboration at multi-site. Okay, so when we talk about high dimensional data, we run into a challenge when we try to characterize it. And so this is a really great parable that kind of goes into that. It's called The Blind Man and the Elephant. And what this is we have scientists located at all different parts of the elephant. Each one of them is going to be blindfolded, and they're only allowed to characterize the object in front of them based on what they're allowed to touch or what they can reach. And so we have this gentleman at the front of the elephant that is reaching out and touching the tusks and is fully convinced that what the object is a spear. Then we have the gentleman at the back of the elephant who's reaching out and touching that tail and is fully convinced that the object is a rope. Then we have someone down by the legs of the elephant, fully convinced that it's a tree because the leg of the elephant feels like a tree trunk. So this is a really great depiction of what ends up happening when we try and characterize high dimensional data using traditional flow cytometry methods. We actually run into this problem because we're not stepping back and looking at the full picture of our data. And it's a really big problem because we would never want to classify our sample as a spear when it's actually an elephant, right? And so hopefully this shows you the challenges that we face when we start to get into higher dimensional data analysis. So we also run into some key challenges, right? So when we're in the low to medium space, there's a few challenges, but it's not too overwhelming. Once we get into this high dimensional space where we're increasing the number of parameters and the number of data points and the number of samples, we run into everything that we have on the right-hand side of the screen. So when we talk about data management, once we get all of these data files, how do we then successfully manage those data files? Where do we store them? How do we archive them? How are they easily searchable? How can I access this data really quickly without having to go to a binder or a lab notebook to figure out the complicated data file path that this data is saved under? And then we talk about data visualization. So once I have my data collected, how can I visualize it so I don't run into the problem that we talked about previously, so that I can properly characterize my data? Result reproducibility. How do I ensure that what I'm doing now and the money and the time that I'm spending is going to be able to be reproduced down the road? We also run into variables in manual gating, so person-to-person variability and population-to-population variability. We also have to think about computational power. So when we're working with these large data sets, our computational power is going to need to increase with the amount of data that we're working at. And then also collaboration. So once I have all of this data, let's say that I want to share it with people. Let's say I find this really, really cool discovery. How can I share that with people? How can I make sure that the community is benefiting from my research? So we also run into challenges in multi-parameter data display. So on the left-hand side of the screen, you'll see all these different bivariate plots that are how we're used to looking at cytometry data. And so even just these plots that are located on the screen, it's a little overwhelming. And we're only looking at something like 4 or 5 dimensions over here. So the figure on the right is showing us as the number of parameters increases, we have to increase the number of plots exponentially, right? So the figure on the right-hand side of the screen only goes up to 16 parameters, and we're already at something like 250 plots. So what happens if we want to take it to the next level and we want to run 20 parameter data? How about 30 parameter data? Do we want to create all of those n by n plots, and do we want to have to use all of those n by n plots to analyze our data? I can barely tell what's going on with 4 or 5 dimensions. How am I possibly going to be able to tell what's going on in 20 dimensions or 30 dimensions? So this is where computational visualizations come into play. So we already talked about what's on the left-hand side of the screen. That's the combination of all of my parameters versus each other. And so how do we reduce the complexity of what we see on the left-hand side of the screen and eliminate the bias of manual gating and the number of the bivariate plots? And that's where these computational visualizations come in. So this is a VisNI run, and what we're doing is we're plotting the VisNI run. And VisNI is a dimensionality reduction method. So what that means is it takes all of the different dimensions of the data on the left-hand side of the screen, and it reduces it down to 2 parameters, TISNI1 and TISNI2. And from those 2 parameters, you can see we have this really nice visual representation of the data. And it creates what we call a VisNI map. We have all these different islands on that VisNI map that we can easily gate and do traditional flow cytometry methods on to classify the phenotypes of those different islands. And what we run into is we have everything on that VisNI map that is portrayed across those n by n plots. So it's going to be much easier for our brains to look at this VisNI map and pick out what's going on across the VisNI map instead of having to decipher what's going on across all those n by n plots. And so Cytobank comes in as a comprehensive solution here, because Cytobank is going to be the cloud-based platform that we talked about earlier. You can see that in the cloud space, we do this high dimensional data analysis. What that means is we use the cloud and the resources and the compute power of the cloud to calculate these computational visualizations. The reason why that's important is because these are really resource intensive on your local computer. So sometimes we have to worry about purchasing a supercomputer just to run these algorithms. The way that it works within Cytobank is we're just going to set it up and we're going to close our web browser, and then we get an email when our algorithm is completed. We can also run multi analysis at the same time. So we can run multiple different algorithms at the same time within the cloud platform. So it allows us to do simultaneous runs, so we don't have to wait for the first run to finish and then go back and re-optimize it. And because we're in the cloud, we can securely save and archive all of the data analysis. So not only the data files will be there, but we'll be able to archive all of the experiments and all of these advanced data analysis techniques that we're using. Once we have everything created in an experiment, we're going to be able to really easily collaborate and share with our colleagues. And another great part about the Cytobank platform is that it was made for biologists. So we're not going to have to worry about coding when we're talking about accessing these algorithms. These algorithms are going to be able to be fully accessed within the platform without having to worry about coding or without having to worry about installing any plugins. So all of that's located within the cloud. So then in addition to that, we also have an informatics consulting service. So those are a couple data scientists that we have on staff here, and they have the background that's necessary to be part of your data analysis and to fill in that gap. So certain institutions will have access to informatics consulting groups, but they're going to have an extremely genomics heavy background. They're not going to necessarily understand the biology and the science behind what's driving the analysis that you're trying to do. And that's the gap that these informatics consulting service fills, because they have that background biology and they can step in and they can help you design an experiment, they can help you analyze your data, they can help you streamline your analysis and create pipelines and write additional code. Whatever you need them for, that's what they're there for. So it's a really great solution for customers who are looking to fill that gap. Okay, so now let's go a little bit into the Cytobank user interface. And so on the left-hand side of the screen, what you'll see is our Cytobank login. So you just go to that in a web browser. You enter in either your username or your email and then your password, and you can log directly into the platform. We have user resources available on that web page, so you can see our how-to guides to get started. We have quick videos, and then we also have recorded webinars available for you. And once you log into the platform, you're met with the experiment manager. And the experiment manager was designed to look similar to an email inbox. And it's supposed to be extremely intuitive for the end user to use to manage and archive all of their data analysis. So on the left-hand side of the screen, you can see that we have all these different filters we can use to access our different experiments. We can filter them based on if they've been shared with me, or if they're my experiments, or if they're Cytobank curated. So that's what we're looking at currently on the right-hand side of the screen. We can also add additional labels to these experiments so that we can make sure that we have them annotated fully as much as we want them to be. At the top, you'll see that we actually have a search bar. So we can come in here and we can use the search bar to search for experiment names or search for different types of experiments. And then in the actual experiment manager, you can see that we provide you with the name of the experiment, and then we have some icons. So the first icon, that little test tube, represents how many FCS files are located within that experiment. And then the additional icons represent the advanced analysis that we can run on these. So Spade, Visney, Citrus, and Flowsom. So it's a really great visual representation to remind us if we've run any of these advanced analysis on our data. The primary researcher is the next column, and that is going to be the person who has created the experiment and the person who has the ability to share that experiment with other people. And then the last column is the updated date. And we can sort and filter on any single one of these columns. So it's going to be really easy for data management, data archiving, because everything's going to be centrally located within this interface. We also allow users to customize access for collaboration. So when you first start an experiment, it's going to be a private experiment, so only you're going to be able to have access to that. Once you start working on your experiment, maybe you want to collaborate with your team and you want to share what you're working on with your team. So you have access to do that. And then you can increase how much you're sharing it into the limited project access view. So we can share this with reviewers, and we can share it with a large project team. And then once we make a really cool discovery and we have a really cool analysis set up and we publish that data, we can make it a public experiment. And that means anyone on your Cytobank server is going to have access to that experiment and is going to be able to view and benefit from that experiment. So you can see how easy it is to collaborate among different groups within the Cytobank platform. We also need to talk a little bit about security, because we're sending all of this data up into the cloud. So any basic and clinical research is going to be secure in the cloud. We use state-of-the-art technology and practices to protect our customer data. We back that data up and we store it at secondary centers, and it's encrypted. We do automated and security scans routinely with Cytobank. So Cytobank is trying to identify any weaknesses that we may have. We're audited by and actively used by leading global pharma and biotech. So we have all of these security measures in place to ensure that your data is going to be kept securely on our cloud. If you have any additional questions, we actually have a security white paper that details all of our security practices. You can reach out to us at cytobank_support@beckman.com, and we'll be sure to forward you along that security paper. Okay, so the next thing we're going to talk about is the API. So the API stands for Application Programming Interface. And what this allows us to do is it allows us to extend the functionality of Cytobank outside of the cloud. So we have all of our data and all of our analysis stored in the cloud. The API can come in, and the API can automatically grab maybe FCS files from the cloud, bring them down on my local resource, and run a custom pipeline on those files. And then once it's done with that custom pipeline, send those data files back up to the cloud for data storage and data management. So it's a really, really cool application. The other thing we can do is we can send data out to an LIMS or central repository. And so the API just allows us to extend the functionality outside of the cloud and continue to build what we need to build to further our data analysis. These are the algorithms that are currently integrated into the platform. So Spade, Visney, Citrus, and Flowsom. We have 4 algorithms currently, and all of these are going to be fully integrated into the platform. You're not going to have to worry about coding. You're not going to have to worry about installing any plugins. You're just going to be able to load in your data set, adjust the settings based for these algorithms, and then click Run. So we're going to dive a little bit deeper into the algorithms. The additional boot camps are going to do deep dives into each one of the algorithms, but this is supposed to be a basic overview. And the first one we're going to talk about is Spade, spanning tree progression analysis of density normalized events. And you can see here is what my Spade tree looks like. So Spade is actually a clustering algorithm. So it takes cells that are phenotypically similar to each other and it clusters them into clusters. And then it maps them out on this minimum spanning tree. And it connects each cluster together and it creates this really great visual representation of our data. You can see we can clearly identify all these different types of cells just by briefly looking at this. It's going to be color-coded based on expression. So anything that's going to be that red color is going to be positive. Anything that's the blue color is going to be negative. So we can automatically identify populations just by looking at a Spade tree. The next algorithm is TSNE, also known as Visney within our platform, since we're doing an approximation of TSNE. And that stands for T-distributed stochastic neighbor embedding. And TSNE is actually a dimensionality reduction method. So what that does is it takes all of your different dimensions of your data and it reduces it down to 2 dimensions, TSNE 1 and TSNE 2. And what that means is it creates these really great visual representations of your data. So these are Visney maps that we ran, and you can see that each parameter has the same identical Visney map, except it's color-coded different. So the color coding is going to help me identify expression among these different islands. And so now I can really easily identify co-expression across my entire data set. And you can see, for instance, that on the CD4, we have a really bright red island, and that means that that specific island is positive for CD4. And then we can look across all of our different parameters and see co-expression. So it's also positive for CD3, and it's also positive for CD45. So that's the way that we can use VISNI to help us further our data analysis. It's going to allow us to really easily identify interesting and rare biological subsets. The next algorithm is Flowsom. So what Flowsom is it's also a clustering algorithm similar to Spade, except it's going to be much, much faster. It also has additional features available to it. So in addition to just doing regular clustering, Flowsom is actually going to meta-cluster the data as well. So you can see that we have all of these individual pies, and then some of the pies are grouped together and color-coded. So the grouped together and color-coded section is a meta-cluster that Flowsom determined. And so this is going to allow us to reveal how markers are behaving across all of our cells, and we're going to be able to detect subsets that we may have otherwise missed. It's going to give us a great visual representation of what's going on within our data set. We can also take this Flowsom and layer it back on top of a VISNI. So the two in conjunction are going to be really useful. And it's going to allow us to automate our data analysis a little bit more. And then the following algorithm that we have fully integrated into the platform is Citrus. So Citrus stands for Clustering Identification, Characterization, and Regression. And Citrus is going to be really useful when we're talking about biomarker discovery, because what it's going to do is it's going to take some user input. So the user has to identify different groups within their sample. So in this specific example, we're looking at a reference group and a stimulated group, and it's going to determine statistical significance between those two groups. And so it's going to allow us to predict biomarkers, and it's going to help us accelerate scientific discovery, right? So the output of this on the left-hand side of the screen, you can see that we have a Citrus output. It looks a little similar to what we're used to seeing with Flowsom and Spade, except it's a hierarchical tree. So each parent cluster contains the children and the grandchildren. But it identifies different parts of the tree that are statistically significant by making them red. And then the output is to give us this great visual representation of the breakdown between the reference and the stimulated group across one of our biomarkers. So it's really going to be useful when we're talking about using this in basic and clinical research. In addition to being able to analyze FCS files that come from flow cytometers or from mass cytometers, Cytobank can also handle additional data types. So what we can do is we can use what we call the Cytobank Drop to take any tabular data and convert it into FCS format. So tabular data here that could be applicable would be site-seq data. Maybe we have imaging data that's in a tabular format. So we can take it from, for instance, a CSV file, insert it into Cytobank Drop, and Cytobank Drop is going to convert that to an FCS file. Once we have the data converted to an FCS file, we can leverage all of the machine learning tools to do analysis on these different types of data files. So at the bottom of the screen, you can see we can look at site-seq data files on traditional flow plots. We can also run VISNI on them as well, and we can do Flowsom, Citrus, Spade, so on and so forth. So the Cytobank Drop is a really cool application that we can utilize in order to continue our analysis on different file types. So now we're going to talk a little bit about the workflow from raw data to insight. And so this is kind of the step process that we have to follow. And when we talk about running these algorithms, we need to make sure that we are still paying attention to the data pre-processing. So all of kind of the techniques that we're used to using in our data analysis still come into play when we talk about running these data visualizations. Junk in equals junk out. So the data pre-processing step is actually extremely important when we're talking about high dimensional data analysis. And what do we mean when we say data pre-processing? We need to make sure that the data quality is good. So we're gating to remove junk and debris from our sample. We need to make sure our data scaling is correct. So all events are going to be displayed on our axis. And if we're running traditional flow cytometry data, we need to make sure that we're doing compensation correctly. If we're running spectral cytometry data, we need to make sure that we are correctly unmixing our data. And so all of this can be done within the Cytobank platform on the cloud if we would like to go that way. But Beckman Coulter actually just released a plugin for Kaluza that allows us to do all of this data pre-processing locally in Kaluza and then upload the pre-processed data files to the cloud to do all of our visualizations then within the cloud. So we have the flexibility here to make a decision if we want to do everything within Cytobank or if we would like to use Kaluza here as part of the process. So what I'm going to do is I'm going to show you how to utilize Kaluza for data pre-processing. So I have a layout created within Kaluza. And I just did some very basic gates. I did a living cells gate, just a cells gate. Now let's say I want to apply a gate here on my single cells. I have the ability to just come up here, select a polygon gate, and create that here. And then I can actually come in and change the gate name. So on my gate, I can come in here and select single cells, let's name it. And if I want to change the color as well, I can do that so maybe it's not so bright. Okay, so now I have my single cells gate. The other thing that I can do in addition to data pre-processing and gating these things is I can double check my compensation. So compensation within Kaluza is very, very easy. I can select a plot and I can just come up here and select my compensation plot sliders. And once I have my plot slider selected, I can come in and I can adjust my slider bars to adjust the compensation and make sure that the compensation is being done correctly on my sample. And then I can also come in here and use the scaling bars to adjust my bi-exponential transition point here and make sure I have my data scaled properly before I send it up to the cloud. Once I have my data ready to go, what I can do now is I can go up to the plugins tab and I have a Cytobank plugin. If I click on that, it's going to prompt me to log in with my username and password. So I'm going to go ahead and do that here. And then I can just hit the login button. And it's going to log me into my server. It's going to allow me to choose the data file I want to send up to the cloud and the gate. So if I want to run an algorithm just on my single cells, I can go ahead and select that here. I can include the compensation matrix and I can also apply the logical transformation. So that means all the data scaling that I did locally will be exported directly up into the cloud. So I can use that for visualization purposes once I'm within the cloud platform. I can also come in here and modify my keywords. So if I'm working with any clinical human data, what I can do is I can de-identify it from this window. If I click on the green plus sign, you can see I can edit keywords here to de-identify. And then I just click on the next button and it's going to prompt me to save this data file. And I'm just going to call this single cells now since I'm exporting just the single cells here. After I hit next, it's going to take me to my experiment manager locally on my computer. And you can see it's going to show me all of the different experiments that I have, the created date, the update date, so on and so forth. So I can click on this create if I want to create a new experiment within the cloud. And I can name this Kaluza test. Define a purpose. And then click save. And these purpose and comments can be anything that I want them to. Now you see I have an experiment created within the Cytobank cloud from my local resource. I can click upload. It's going to ask me if I want to make sure that I want to upload my FCS file into that experiment, and I click yes. And then it's going to upload this data file into the cloud. Once I have this data file uploaded into the cloud, I'll be able to do any additional advanced algorithms on this data file. So this is the pre-processed data file that's already gated. So I just have my single cells gate. So now it's ready to just process an algorithm on that data. And I have the option to do this within Cytobank, but I also have the option to do this locally if I would like. So this just verifies that the upload was successful. So now what I can do is I can come up into my internet browser. I can log into my Cytobank Premium account. When I log into my account, you can see I'm met with that experiment manager. This is the new Kaluza test experiment I created locally, and you can see that I have one FCS file located within that. If I click on it, it brings me to my experiment summary, and you can see it uploaded my data file directly into the cloud from my local resource. Okay, so I'll go back to Kaluza now. I'll click OK, and I'll exit out of this. Additionally, I can also bring data files down from the cloud after I've run the algorithms on them. So if I want them to come back locally, I have the ability to do that. And then I can use Kaluza to create a layout or a protocol that works for me to analyze the data locally if I want to do that. So you can see here, this is a VISNI that was run on this data. This is 20 color data from the Cytoflex. And what I can do is I can create a gate on a t-SNE plot, and I can show that gate backgated on a plot here where I'm looking at CD3 versus CD16. So you can see this island here represents my CD16 positive, CD3 negative population. And what I can do is I can move this gate around on my t-SNE plot, and you can see as I move it around to each one of these different islands, it's going to backgate and change the color on the population. So this specific island down here is going to represent the CD16 positive, CD3 lows. And as I move it around the screen, I'll see the color update in real time. So it's kind of a cool functionality that I can do. Additionally, when I was within Cytobank, I ran a VISNI and a Flowsom. So I have the ability to come down here and also look at my Flowsom analysis. So this was a VISNI run, and I also ran Flowsom. So the Flowsom came out with all of these different meta clusters. So what I did was I placed a gate on each one of these meta clusters, and then I backgated that onto the t-SNE map. So you can see all the different populations from the Flowsom meta clusters are color coded over here on my t-SNE map. And so I can break it down into different subpopulations. You can see with the Flowsom, it's gonna cluster automatically. So it's going to break out populations that maybe even I visually wouldn't have been able to. So for instance, this island is going to represent a different phenotype than this island. And then I can take all of these different colors and I can show them on that same CD3, CD16 plot that we were looking at on the previous screen. And I can see where they all show up. So this is ungated. It's representing every single population. If I want to hone in on one specific meta cluster that Flowsom came over, I can come over here and I can visually look at cluster number 6, and I can see that that is that population up here that's CD16 positive. If I want to focus in on cluster 4, I can do that, and so on and so forth. So I can come in here and I can change all the different meta clusters that I'm looking at. Okay, so now we're going to go back into the PowerPoint. And so in summary, classical data analysis approaches may not be sufficient for more complex data sets. So collectively, Cytobank and Kaluza can provide a workflow solution that's going to fit our needs. So we can do integrated data QC and pre-processing workflows. We can do that either within the cloud, within Cytobank, or we can do it locally in Kaluza. We have a variety of machine learning algorithms available that don't require any coding or any plugin installation. We have secure data management options available for you within Cytobank, so you can access your data anytime, anywhere. Especially during a time like this, it's going to be extremely useful to be able to access everything that you've been working on. We're going to be able to do all of this for complex, high-dimensional data sets that are critical in understanding and progressing our scientific knowledge. To get started, you can visit our website and sign up for our free 30-day trial. Additionally, you can take advantage of our training and consulting services. We have educational resources and our support and help portal. You can reach out to us, cytobank_support@beckman.com. We're also offering additional office hours if you want one-on-one time with a staff member. So you can access that through the support portal. And then we also have our upcoming bootcamp sessions. So on April 16th, we're going to be doing the Cytobank platform in-app demonstration. April 21st will be dimension reduction with Visney. April 23rd will be clustering with Flowsom. April 28th will be biomarker discovery with Citrus. April 30th will be apply machine learning algorithms in Cytobank to non-cytometry data. So these are going to be great to use to learn the platform. So thank you so much for your attention and your time. This is one of my pups who helped today with the presentation. We hope you appreciated it, and we'll open up the sessions now for a Q&A. And we look forward to seeing you at the additional boot camps that we'll be running in the month of April. Thank you so much.